Good afternoon. Thank you everyone for joining this section. My name is Wu-Fu Chen from Taiwan Kaohsiung Chang'e Hospital, and my co-moderator is Professor Sato from Osaka Kansai Medical University. Professor Sato, say hello to everyone.
Okay, thank you. I'm Takanori Saito from Kansai Medical University in Osaka, Japan. It's my pleasure having being the chair of this session.
This session will discuss the application of IOM in spinal surgery. Today's first topic is the basic science of brain stimulation in IOM of spinal surgery. It gives my great pleasure to introduce Professor Lo from Singapore National Neuroscience Institute to this wonderful speech. Professor Lo graduated from the National University of Singapore and completed his postgraduate fellowship in clinical neurophysiology at the University of Oxford. He was the previous head and the current senior consultant neurologist at the Singapore National Neuroscience Institute and the professor of Duke and U.S. Graduate Medical School. Professor Lo is active in the field of clinical neurophysiology and lecture regular on this subject in the world. He was the founder president of the Singapore Clinical Neurophysiology Society. His main research interests are on the spinal cord disorder. He is the concurrent editor board of clinical neurology and neurosurgery, and he is the regular reviewer of over 30 international journals. Let's welcome Professor Lo with the warm applause. Professor Lo, please.
Greetings. The topic I am lecturing today is on the basic science and basic principles of brain stimulation for the intraoperative monitoring with regards to spine surgery. The next slide will show my disclosures and sources of my support. In today's talk, I will introduce the subject and briefly run through the objectives of spinal cord monitoring in the operating theater. I will focus on four aspects of basic science. First, the functional neuroanatomy, followed by fundamentals of non-invasive cortical stimulation, including historical aspects, and physiology with regards to the central and peripheral augmentation of motor evoked potential amplitudes. Finally, I will sum up on what is the current IOM strategy, conclusions, as well as moving ahead, what is the future direction. As you know, IOM, or intraoperative monitoring, is most necessary to ensure safety of an operation. And we are talking about using an electrophysiological technique, and it ensures the integrity of the nervous system, where structures at risk are subjected to trauma. But the ideal IOM technique should thus be reliable, specific, with minimal interference with the operation. It should be safe, cheap, and low in manpower costs. This may involve more than the use of standard electrophysiological techniques, as you can see. IOM is a team effort comprising not only the surgeon, but the electrophysiologist or the technical staff, operating theater supportive staff, anesthetists, technicians, as well as other vendors. The IOM environment is obviously hostile, and also there is a lot of constraints in terms of space. There are chemicals, anesthetic agents at play, and there are obviously human and emotional factors in this high-stress environment. The types of spinal surgery we're talking about today involve cervical spine surgery, degenerative spine surgery, tumors, scoliosis in which the patient is neurologically normal, technically, as well as surgery for other spinal cord disorders, including shrinks and aneurysm. So the scope is very large. Working against this is the depth of the anesthesia and maintenance of vital signs. The intraoperative blood investigations and imaging can also be considered as part of intraoperative monitoring repertoire. The risk of the spinal cord injury can be manifold. It can be susceptible to global injury, but in animal studies, selective injury models can be demonstrated. The processes include ischemia, compression, blunt trauma, blood pressure changes, vasospasm. For somatosensory evoked potentials, the dorsal column is largely monitored, whereas in motor evoked potentials, or MEP, most of the monitoring is in the corticospinal tract, but as you can see later, other tracts could be involved. There are certain vulnerable areas where vascular injury is particularly important, and these include the watershed areas. In SSEP, unfortunately, the monitoring is in the dorsal column. Stimulation is via peripheral nerve in a fairly fixed type of stimulation protocol, and averaging is involved. So there is a lack of instant feedback with regards to changes in the somatosensory evoked responses. It is well known that SSEP is often less efficacious compared to MEP, although it has complementary value. So this shows you the SSEP of a patient that we monitored in real time, but each response is actually a composite of multiple averages. Spinal cord evoked potentials is another way of looking at measuring motor and sensory responses. It can be cord to cord. That means we are talking about stimulation of the cord and recording from the cord, or stimulation can be by means of an epidural catheter. This method has been described by Dr. Tamaki, and it involves a response that is actually comprised of an initial spike and polyphasic components. It is touted to be less susceptible to drugs, but it is invasive, requires averaging, that means lack of real-time feedback. And if stimulation is cortical, then D and I waves monitoring can be picked up using the epidural catheter. This I will elaborate slightly later. So this is the catheter that we're talking about, the epidural catheter that is used invasively in the epidural space to pick up spinal cord evoked potentials. This method is currently not used very widely. Before I go into the different methods of brain stimulation, let us revise the functional neuroanatomy. The functional neuroanatomy is an important part of the basic science in IOM of spinal surgery. We often talk about stimulation of the motor cortex, whereby the corticospinal tract is excited and there are descending volleys that summate, cross at the brainstem and summate at the anterior horn cells. The muscle responses are called MEPs. But we mustn't forget that the spinal cord also consists of other descending tracts, including the medullary tracts, medullary reticulospinal tracts, and other smaller tracts that may be less cross and descend ipsilaterally and contralaterally. This includes vestibular spinal tract, tectal spinal tract, and reticulospinal tract, amongst the other tracts that we know. And these tracts are often forgotten during IOM. This is another diagram to show you that the anterior and posterior and lateral cords have many different tracts that descend in an ipsilateral and cross fashion. So the descending motor pathways, particularly when it comes to motor control, consists largely of the corticospinal tract, which basically process skilled motor activity in the distal muscles, like the small muscles of the hands and feet, as well as the rubrospinal tract and reticular and cortico-reticular fibers in the reticular spinal pathways. These tracts are more important in the postural control of proximal musculature in the maintenance of posture. During electrical stimulation of the motor cortex, multiple descending volleys are needed to achieve optimal stimulation. But we know from basic science studies that electrical stimulation itself of the motor cortex is able to elicit deeper structures. And even in animal studies, stimulation can be down to the level of the pyramidal decussation and maybe even deeper. In montages involving anode and cathode, where the vertex and the midline respectively are used, deep activation of descending cortical spinal tracts in humans produce multiple D-wave volleys. By means of DTI or DTT, diffusion tensor tactography in MRI, we're able to show that even in children, these projections span from the premotor area down to the corona radiata internal capsule and descend very close to the reticular formation. These corticular reticular projections mature at different rates compared to the cortical spinal tract, although they descend very closely. There are some implications with regards to the developmental pathophysiology of scoliosis. So these cortical reticular fibers project from a very large area in the sensory motor cortex down to the reticular formation of the brainstem. The precentral gyrus origins project to neurons in the pons and medulla in the form of the lateral and medial reticular spinal tracts. The sensory motor cortex projects to the paramedian and reticular nuclei of the reticular formation in the brainstem, and this supplies also the cerebellum and enable the cortex to make influences on motor function at many levels, including the spinal cord, as well as the cerebellar projections. The long ascending fibers projections of the reticular formation or the ascending reticular formation have produced a different function. And this is important in keeping awake and cortical arousal. The cortical reticular projections are important in evoking EPSPs in pontine and medullary reticular nuclei. In animal studies involving monkeys, direct reticular formation recordings can be performed. We also need to know that the cortical spinal tract, which descends next to the reticular spinal tract, can also be uncrossed, and in some studies up to 30% can be descend ipsilaterally for the cortical spinal tract. Also, cortical spinal tracts also have collaterals which project to reticular spinal nuclei, and this causes a much more complicated network of descending motor tracts. This diagram shows you the cortical reticular pathway as well as the cortical spinal pathway as it descends, and you can see that the cortical spinal tract often crosses more at the medulla compared to the cortical reticular tract or the reticular formation, which ends in the reticular formation in the brainstem. This is another diagram to show the actual location of the cortical reticular fibers as well as the cortical spinal fibers. So a diagrammatic representation of what I have mentioned earlier on is that these cortical reticular fibers receive projections from the premotor area and motor areas and descend before synapsing at the reticular formation in the brainstem, the pons, and the medulla, and finally descending into the spinal cord, lateral and medial reticular spinal pathways that eventually control spinal motor neurons. These are important in maintaining posture and proximal muscle tone. So this is another diagram to show you the relative positions of these tracts and the relative positions of these tracts as they connect with the spinal pathways and spinal reflexes in each level of the spinal cord. In the next section, I will cover the fundamentals and historical aspects of non-invasive brain stimulation. The mainstay of non-invasive brain stimulation or percutaneous brain stimulation is the use of electrical pulses to non-invasively stimulate the brain through the intact skull. And it's only in 1980 that Morton and Merton published this important paper that showed how this can be done percutaneously. This must be quite a painful procedure, but it actually succeeded in evoking a motor evoked potential in the hand with a latency of 16 milliseconds. This paper was published in Nature and is one of the earliest to show that one can successfully, in a awake person, manage to evoke a muscle twitch response in the small muscles of the hand. These are many ways to record the motor tracks. During intraoperative brain surgery, you can actually use great electrodes which contact the motor cortex surface, as well as make recordings and stimulation. Earlier on I mentioned in the cord, or make recordings using an epidural catheter where you can record D and I waves, and of course MEPs or muscle twitch recordings in small muscles of the hand and feet, which largely pick up cortical spinal pathways as well as other responses from smaller tracts. There are other methods to electrically stimulate the cortex, but some of them can be at the subcortical level, or it can be stimulated using magnetic flux by transcranial magnetic stimulation. But this method is not suitable under anesthesia conditions in the operating theater. So what is the MEP, or motor evoked potential? It is a muscle twitch response which is evoked by stimulating the motor cortex. It can be recorded as D and I waves in the epidural space, but the corollary muscle response is called a motor evoked potential. This can usually be achieved by an external or inbuilt stimulator in most IOM machines. And MEPs can be evoked using single or train stimulation. A typical way is to deliver a train of 0.5 milliseconds duration up to 5 pulses of interstimulus interval 3 to 4 milliseconds and make recordings in the muscle. This usually results in a summation of descending volleys and finally evoking the muscle twitch. The real challenge is to balance the use of anesthetic agents and neuromuscular blockers, and this is where the challenge of overcoming anesthetic constraints in the operating theater is often seen as an obstacle to successful motor pathway monitoring. So what are D and I waves? D and I waves are often elicited by either an stimulation of the exposed cortex or percutaneously using electrical stimulation. They are often not successfully elicited using magnetic stimulation. The first wave or D wave is usually a direct excitation of the neurons of the cortical spinal system, and the subsequent I waves that follow about 1.5 milliseconds are indirect depolarizations by interneuron recurrent depolarization in these cortical connections inside the pyramidal motor cortex. These multiple descending volleys summit at the anterior horn cell. Although the first D wave may not bring to fire the MEP, subsequent summation of the I waves will reach threshold and trigger neuronal firing. This summation may result in a single discharge, or it may repeatedly fire and result in larger MEP, which can be recorded as a distinct muscle change. Experiments have been conducted whereby electrical stimulation can be recorded at the cervical-medullary junction, similar to the epidural recording, where D and I waves can be recorded during electrical stimulation of the motor cortex. These D and I waves eventually summate to form MEPs, and the size of the D and I waves can be varied according to the intensity of electrical stimulation. In the interest of time, I would quickly say that anesthetics not only impair descending volleys of multiple D and I wave summation, they cause generalized depression of the excitability of the motor cortex as well as the spinal cord and motor neurons. And the train of stimulus where we apply is hoping to overcome this lack of excitability, whereby generating descending volleys can cause efficient summation. In technically demanding surgery, for example, resection of intramedullary tumors, MEP alone may not be enough, and a combination of MEP with D-wave and I-wave monitoring may be necessary despite the invasive nature and costs and expertise needed. Whenever there are MEP changes, it is important to quickly recognize and stop the operation and perform saline irrigation, whereby theoretically the washout of extracellular potassium can facilitate recovery of MEPs and mitigate injury to the spinal neurons. Other mechanisms include stabilization of blood pressure, vasospasm, and temperature. Intraoperative monitoring of neurologically impaired patients is extremely challenging, especially when monitoring of MEPs, which are very small at baseline, difficult to record. And D waves sometimes can be absent or heavily desynchronized in majority of cases. So it is necessary to combine these difficult monitoring with intraoperative imaging. This is an example of a type of stimulator that can be connected externally. This is produced by Digitimer. But most intraoperative monitoring machines have built-in stimulators nowadays that can generate voltages up to 1000 volts very efficiently. Finally, I would like to say that ipsilateral MEPs can be elicited in a muscle from ipsilateral stimulation as well as contralateral stimulation. This is because some of the descending tracts that I've mentioned earlier do not cross as much as the corticospinal tract. So for example, a right cortical stimulation will result in a much larger ipsilateral MEP by not only stimulation across the corpus callosum, but by stimulating the contralateral as well as the ipsilateral descending motor tracts, which comprise of the cortical spinal tract, the reticulospinal tract, vestibular spinal tract, and other smaller tracts. Ipsilateral responses have a role in improving the specificity and sensitivity of monitoring, especially in scoliosis patients where they are neurologically normal to begin with. However, centrally, transcranial MEPs can be enhanced by delivering multiple train pulses with pulse width of 0.5 milliseconds and interpulse intervals. However, this may not result in a more efficient MEP amplitude. So changing from the conventional type of MEP to multi-train MEPs can greatly enhance transcranial MEP responses. This is akin to LTP mechanisms, whereby trains of electrical stimulation can result in a much larger response delivered through NMDA receptor mechanisms. Frequencies as high as 400 Hz can be used, although in theta burst stimulation, using transcranial magnetic stimulation, up to 50 Hz is only ethically used. As you can see, the delivery of these pulses of electrical currents will facilitate LTP mechanisms of the NMDA receptors by releasing more vesicles, and alignment of these receptors can also facilitate a EPSP that is delivered. This is a mechanism that has been used as a model to explain multiple train cortical stimulation in MEP augmentation central. In this particular study that we performed, we are able to show that using up to 9 pulses delivered percutaneously in cortical stimulation can result in the augmentation of motor evoked potential size after the decompression surgery in cervical myelopathy. And this supports the fact that a longer train stimulation results in central augmentation of MEPs that were not demonstrable previously with shorter trains of cortical stimulation, in view of the fact that cortical stimulation with shorter trains has not been. As you can see from this diagram, many of the baseline MEP size are augmented after decompression of the surgery, and this can be seen within minutes and hours intraoperatively. And the reason why this is demonstrable is because of the longer duration, the number of pulses, increasing the number of pulses of cortical stimulation where you can deliver using current technology. So central augmentation of MEPs can be done using a long train of up to 9 pulses, or using a double train where you prime or condition the cortex followed by 7 pulses. This double train stimulation is actually very similar, and we believe both these mechanisms result in temporal and spatial summation to overcome anesthetic effects, much akin by the model of LTP that I've explained earlier. The last section I will talk about peripheral augmentation of MEP. Peripheral augmentation of MEP involves the delivery of a tetanic stimulation of a peripheral nerve before transcranial stimulation, and this results in priming of the excitable neurons in an afferent fashion, and this has been shown to increase the size of the MEP intraoperatively. What is actually the mechanism of this method? It has been shown that tetanic stimulation of a peripheral nerve increased the MEP size recorded in both innervated and non-innervated muscles, whereby the MEP or CMAP amplitudes were best augmented when that particular nerve is stimulated. But there is a general increase in the size of the MEP from other muscles that are not innervated by the particular nerve. So this is an example of peripheral augmentation of MEP amplitude. To investigate the underlying mechanism, this group of investigators used F-wave characteristics during peripheral augmentation of MEPs, and their results actually confirm the hypothesis that tetanic stimulation of peripheral nerve may cause remote augmentation of MEPs by increasing the excitability of the anterior horn cells, and this is derived by measuring changes in F-wave characteristics intraoperatively. Peripheral augmentation of MEPs is akin to this mechanism of paired associative stimulation, or PASS, whereby in the awake patient or awake subject, a peripheral stimulation of the nerve followed by a magnetic stimulation of the cortex results in a much larger MEP size as compared to when one is not primed, when the stimulation is not primed by a peripheral nerve stimulation. In summary, IOM is an emerging standard of care in spinal surgery, whereby monitoring of the MEP provides the most direct evidence of motor function. Transcranial electric stimulation activates both ipsilateral and contralateral tracts below the cortex and at the cortical level. Percutaneous transcranial electrical stimulation should optimally activate D and I waves to ensure efficient summation of descending volleys that summate at the spinal motor neuron as a primary aim. The central augmentation of MEPs may partly be explained by what we know in basic science as LTP mechanisms, and peripheral augmentation of MEPs may partially be explained by paired associative stimulation experiments that are demonstrated with transcranial magnetic stimulation. It is my opinion that more clinical and basic science studies are needed to improve the delivery of IOM in spinal cord surgery. Thank you for your attention, and in better times, we hope to see you again, perhaps in Singapore.
First of all, thank you for your comprehensive lecture. We have 5 minutes for online discussion. The first question is from the Professor Lee. Recently, we began to use the focused ultrasound for brain stimulation. Do you think we can use MEP and SEP to quantify the effect of neuromodulation, just like TMS?
Thank you for a very interesting suggestion. My knowledge of focused ultrasound is not great, but I know that ultrasound delivery can be used to generally increase cortical excitability. How much focal it is, I'm not sure. But it can also be used for as a lesion to lesion the studies, create lesions in the brain.
Thank you. The second question is, this might happen in patients with unilateral intramedullary brainstem lesion, because once the signal enters pons, some will travel to contralateral side and ascend to auditory cortex, with account for preserved threshold for that ear. However, BAP of that side will be blocked by the unilateral lesion. This question is from the Professor Yang.
Yeah, I think that one is not for me. That's for the previous speaker. So I would say that focused ultrasound it has a downside in that it generates a lot of heat, and the safety aspects has not been well studied, and therefore it is still early days to allow focused ultrasounds to be used in even in research and clinical and even clinical settings, because it is a method that is fairly new. But I'm sure there are many other novel ways to increase cortical excitability and maybe utilize it in the intraoperative setting, perhaps maybe to overcome the effects of anesthesia in suppressing the motor cortex excitability itself. I think the second question from Dr. Yang is not for me.
Okay, thank you.
Thank you.
Do you have question, Professor Sato?
Oh, yes. The long, Royal Long,
Hello?
this presentation, thank you. You present a few methods of the augmentation in MEP. We have sometimes experience of the anesthetic fail for the lung surgery. What kind of augmentation method do you choose in MEP during the failed phenomenon? Professor
Yeah.
Long?
Yeah. Hi. Can you hear me? Yep. So we prefer to use the long train stimulation because it is convenient and safe, and we have enough experience. We also try to improve MEP by working with the anesthetist very closely, and also we also try to look at the optimal stimulation position. So, for example, if you were to use C1, C2, or C2, C3, C4, sometimes these positions may not be optimal in obtaining an MEP that is already quite small, particularly in patients with very significant spinal stenosis, spinal cord compression, or tumors, spinal tumors. So by working with the anesthetist, by increasing the train of pulses, and also by adjusting the optimal stimulating positions, it is a more convenient method. Of course, we have tried using the peripheral stimulation augmentation as well, but we find that by adjusting the actual number of pulses, you can actually control the size of the MEP more conveniently, and also perhaps safer in our opinion, because we are more used to using this method.
Okay, thank you.
Okay, because time is limited and so we will proceed to the next topic. Thank you for Professor Lo.